Thanks, Bob. So it's my pleasure today to introduce Eric Crandall. Eric got his BA in integrative biology at UC Berkeley before moving on to grad school, where he did an NSF GK-12 fellow that many of the EECB students of the past will recognize. That involved his interaction sort of with his PhD project, which was on gene flow of Indo-Pacific species across Coral Triangle with Paul Barber. He did a lot of work in Indonesia and the Coral Triangle. That led him to a postdoc with Kent Carpenter at Old Dominion University on an NSF PIRE project that was focused on biodiversity in the Coral Triangle. And then he did a slight detour to National Marine Fisheries, the Southwest Fisheries Science Center for a few years, and decided that he wasn't as excited about salmon as he was about Indo-Pacific biodiversity. And he and I had been working together on this project, the diversity of the Indo-Pacific Research Coordination Network that Eric led, and he decided that he would become the project lead on that. And he was doing that for a while until he accepted a faculty position at Cal State Monterey Bay, which, if you're familiar with the Cal State systems, involve an awful lot of teaching. And that ultimately led to him leaving Cal State and joining Penn State, where he is able to focus a lot more on his research now. And that is what he is going to talk about today, is some of the experiences that he's had in terms of the importance of understanding data and metadata and open data standards that have allowed these projects, and I believe he's going to talk about a couple of those today, how sharing data allows you to answer questions about biodiversity that are beyond the scope of what anybody could possibly do as a single project. And so it's a great pleasure to turn that over to Eric, and please join me in welcoming him. Eric.
Thanks, Rob, and thanks for inviting me out. I'm wearing this Hawaiian shirt. I'm coming to you from the central Pennsylvania, and wearing this Hawaiian shirt is kind of showing that I really wish I could be at University of Hawaii to give this presentation, and also want to make sure that you understand that I'm not wearing this in support of the Boogaloo movement. Just really excited to be invited to Hawaii. Let me share my screen. All right, so I'm going to diverge a little bit from the standard academic talk, just a little bit, in that I'm going to first talk about a little bit about DipNet as an organization and as what we did with the RCN, and then I'm going to give you about 5 minutes for questions and to refresh your drinks or go to the bathroom or whatever you're doing since we're all at home. And then I'll proceed to then talk more about some of the science that I've done under the auspices of DipNet and working with Rob as well. I'm also going to throw out a couple easy questions, so have your hands by your mute buttons, and hopefully people can jump in with some answers as I go. Okay, so I am going to be talking about what we've done with this DipNet RCN. Just start off with a picture of me as an undergraduate at UC Berkeley. This is me at the UC Gump station just south of you in Moorea. And what I'm looking at through the microscope, I actually worked with Richard Emlet and Amy Moran up at OIMB the summer before, and I got really into larvae. And when I got out there, I was looking at this little amphibious larva from a snail that the hi'iwai that you might know of in Hawaii, the amphibious snails that I showed with my undergraduate work were obligate marine dispersers. They have to be exposed to salt water to develop. But there I was in Moorea, and I'm just thinking about the size of that larvae and then the size of the ocean and the size of the Indo-Pacific. This is a region that spans the circumference of which, sorry, the diameter of this region spans about 2/3 of the circumference of the globe. And I had this very subtle observation, which is that the Indo-Pacific is a big, big place. And if we're thinking about marine species, those marine species, in order to maintain species integrity, have to somehow have gene flow across that region. And a lot of species do have ranges that span all the way across the Indo-Pacific region. And what is the way that they do that? This is my first little easy question. How do these species maintain gene flow across? I guess I'm pointing this at the graduate students or undergrads that are in the audience.
Currents.
Currents that carry
larval dispersal.
Yeah. Larvae, yeah. So, and that's what I ended up doing my dissertation on, was trying to understand where and how far these larvae travel on these currents. And at the same time, even though there are species that have the ranges that span this entire region, the Indo-Pacific is also, especially at the heart of the Indo-Pacific, it hosts the highest biodiversity in the entire world ocean. And this is just a map from Roberts et al. 2002 showing the hotspot that we've come to call the Coral Triangle, right there in the Indo-Malay Philippines archipelago. And so a motivating question, I'm not obviously the first person to observe that this is the highest area of biodiversity. Charles Darwin at Christmas Island was rowing across the ocean on a clear day. Obviously, he didn't have scuba gear. And he wrote in his typical sort of Victorian phrases, "It is excusable to grow enthusiastic over the infinite numbers of organic beings with which the sea of the tropics, so prodigal of life, teems." And then Alfred Russel Wallace, of course, spent quite a bit of time in that archipelago. And he was looking down, again, obviously didn't have as much of a chance to study these organisms as he probably would have liked. But he wrote, "The clearness of the water afforded me one of the most astonishing and beautiful sights I have ever beheld. The bottom was absolutely hidden by a continuous series of corals, sponges, actiniae, and other marine productions of magnificent dimensions, varied forms, and brilliant colors. It was a sight to gaze at for hours, and no description can do justice to its surpassing beauty and interest." So it's fair to say that a motivating question for me and for many others, including Rob and Brian and others, is how did the Indo-Pacific become so diverse? And yet, even though it's the largest marine biogeographic region in the world and the most speciose, it ranks 5th. And I did a fairly generous search here. I searched for marine biology, ecology, or evolution, and these various regions of the world ocean. And it came in 5th in terms of numbers of studies that have been done on it. Again, for the graduate students in the audience, any ideas why it might have come in 5th?
Would it be because the fisheries aren't really like as important as other ones like on the east and the west coast?
Well, sort of along those lines. I mean, another clue would be, why is the North Atlantic so well represented?
Is it due to access?
Access and, well, more specifically to funding. For marine ecologists who are well funded in countries like the United States and Europe, they can go in their backyard and study the North Atlantic all they want. For marine ecologists in the Indo-Pacific, mostly coming from developing countries, They simply don't have the funding to create the same amount of science. And so there are numerous challenges for the studies of Indo-Pacific, and I'm just going to focus here on molecular ecology and evolution. The first set of challenges are logistical. First of all, it's just its sheer size. Second of all, its diversity, its political complexity, and the general overall lack of cooperation that has occurred, at least in the past. There are also numerous theoretical challenges, and I'll go into these in the second half of my talk, but low values for FST estimators, large effective population sizes and high genetic diversities, and the larvae creating these potentially high migration rates. So for this first half of the talk, I'm going to focus on DipNet as a research coordination network and what we've done to try to overcome these more logistical issues. Then we'll take a little break, and then we'll come back and we'll talk about some science and some things that I've done to address these particular problems. All right, so again, just to outline the problem in a little bit more detail, the size of the Indo-Pacific is a huge problem. It's an opportunity, but it's also a problem in terms of how do you sample, if you want to understand the phylogeography of a single species, how do you sample across its entire range without getting a huge grant and flying all over the place? Its diversity is, again, it's an opportunity, but it's also a challenge. How do you, in phylogeography studies of other continents or oceans, people pick exemplar species and they find patterns and they say, okay, well, this species is a great example for all the vertebrates, and it's much easier to understand. The diversity of the Indo-Pacific makes it very difficult to make sense of it all. The political complexity, again, the size of the Indo-Pacific makes it impossible for, I couldn't, in searching the internet, I couldn't find a single map that showed the exclusive economic zones of every country in the Indo-Pacific, but there are at least 65 countries whose exclusive economic zones intersect with the Indo-Pacific. And each of those countries is a signatory to the Convention on Biological Diversity. They have their own permitting process. And that, again, that also makes working across the Indo-Pacific a very politically difficult thing to do. And then in the past, there's sort of been a lack of cooperation. This wasn't so much because different groups were competing with each other so much as they were trying not to step on each other's toes. So this is a area cladogram of, it's like an area cladogram that you would do in a biogeographic study where you're looking at similarities between taxa or between flora and fauna across different areas. But this was actually just for species for which we had genetic samples. So it's more a comment on what has been studied than what actually exists there. And what you can see is in blue is my doctoral lab, Paul Barber's lab. We had our own little group of species that we targeted when we went there to sample. And so everything in blue is what we studied. Then you've got the Tobo lab out in Hawaii. You've got the French in French Polynesia and Réunion, and you've got the Australians working on the Great Barrier Reef. And then in purple, you've kind of got everything else that's been done in the Indo-Pacific. And it just shows that there's very little overlap in the terms of the species that were selected by all of these different groups. Very little co-sampling or cross collaboration across these different groups that are all working towards the same questions. And in addition to that, a lot of us, and I'll include myself in this, we were doing this in sort of a parachute science style. We were flying into these countries, getting our permits, getting our samples and then heading back to the United States or Australia or some other developed country to do our molecular work and our analyses, and then publishing. And we weren't necessarily including scientists from Indo-Pacific countries in a real collaborative fashion. Maybe we were putting them on as authors or something like that, but we weren't truly developing collaborations and moving their science forward, or helping them to move their science forward as much as we were moving our science forward. So in 2012, I got in contact with Cynthia Reginos and then Rob, and we got a nascent catalysis meeting to bring together all the researchers who'd been working in the Indo-Pacific. We wrote a special issue in the Bulletin of Marine Science, and then we got a working group grant funded for meetings following that, all at Nescent, and formed a nice little group of friends that we really have worked well since then. These include Libby Liggins. I don't have a picture of Chris Bird in here. Michelle Gaither, Rob, Cynthia. That's really been so far the core of the DIFNet group. We took what we had done in the working group, and we had put together this huge database of that I'll talk about later, of our of genetic data from the Indo-Pacific. And we wrote a NSF RCN, which we were surprised and happy to find got funded. And that funded 4 workshops for capacity development in the Indo-Pacific, DIPNet coordinator, who was me for a little while. And we developed memberships, over 70 members from over 20 countries in the Indo-Pacific. Our goals were to further develop this open access genetic database that would, our goals for that were that, a researcher or a graduate student in the Philippines is not likely to be able to get funding. If they might be able to get funding to sample their critter from around the Philippines, but they're not going to get funding to sample outside of the Philippines. And our goal is to make all of the data that was around available and openly available to them, so that they could put their data into the greater Indo-Pacific context. We wanted to promote strong collaboration and data sharing, and then we had a couple of some fundamental research topics that I'll talk about in the second half of this presentation. So we ended up putting together a database of over 35,000 mitochondrial sequences from 200 species across the Indo-Pacific. This just shows the distribution of species and sequences across the region. And I happened to end up talking to this guy, John Deck, who had been developing something that he called the Field Information Management System. And so this is like a laboratory information management system, but his big point was that samples don't originate in the lab, they originate in the field. And we should start tracking their metadata as soon as we get that sample onto our boat. And it should get a number right then, and it should be entered into a spreadsheet, and the metadata should be tracked starting from there into the laboratory and beyond. And so we ended up, our database that we had put together ended up sort of being the first use case for what ended up being in collaboration between us and John as the developer, this Genomic Observatories metadatabase, or GEOME. So it's open access, and we're focused on the metadata. Importantly, we're not trying to reinvent the wheel of storing genetic data. That's already done very well by GenBank, or now it's more properly referred to as the INSDC SRA. But we just wanted to keep the data that describes all of the who, what, where, when of metadata that are associated with these samples and with their derived sequences and genotypes. Again, we're not trying to reinvent the wheel. We create a permanent link to data that are stored in GenBank and that are blastable. And we just store the metadata, which, to be honest, GenBank doesn't do all that good of a job of, if you've ever tried to submit sequences to GenBank, you might know what a headache it is to try to get your lat longs and your dates and all the other important metadata that you'd like to be attached. It can be quite difficult. In developing this database, we followed the genomic and biodiversity metadata standards that have been developed by the Genomic Standards Consortium and Darwin Core. So, you know, if there's probably 10 different ways that you could label a field for longitude, but the Darwin Core name for longitude is decimal longitude. And if everybody uses that standard, then we can agree and our data can be interoperable. A really nice feature of Geom is that we allow communities to adopt their own data standards. So if we view DipNet, or the group of researchers that comprise DipNet as a community, we came up with our standards for what we thought should be attached, what metadata we thought should be attached to genetic data. So we all agreed that we thought the principal investigator should be a field that is associated with every sequence that comes out of that principal investigator's project. So that was one field that we thought was a requirement. Other communities, like let's say a community of people who are sequencing the microbiome of cow guts, maybe they have their own set of standards that's very different than a group of population geneticists in the Indo-Pacific. They can determine their set of metadata that they think are important. There are a few minimum fields that are common across all projects. But by doing this, we sort of allow flexibility among different research communities. I worked with an international, a lawyer on international law to create a data usage agreement that addresses the Convention on Biological Diversity. I won't say much more about it than that, but if you're aware of the issues around the Convention on Biological Diversity, then I think we've figured out a way to address those. We've also developed an R package that allows you to search the geome metadata. So if you're interested in all of the echinoderms from Papua New Guinea, you can search the geome metadata for those samples and that's in one command. And then in a second command, you can use the SRA toolkit to batch download all of the data that were created or that have been accessioned to Geom that also have the associated genomic data in the SRA. We have citable data publications, and keep an eye out for a paper coming out in Molecular Ecology Resources. It was just accepted, and they'll sort of be endorsing it, and they're looking to start recommending it to submitters to molecular ecology, with moving towards eventually requiring it. So we'll see how all that works out. And then I should say that I was supposed to be in Hawaii in August as our last meeting under DipNet. Obviously, I couldn't travel, and instead we turned that into a datathon. So we took students who had lost income, graduate students who have lost income from the pandemic, and we hired them to do a little datathon where we surveyed over 1,600 bioprojects that were potentially relevant to natural populations, but genetic diversity of natural populations. We don't have the full results from that datathon yet, but we did determine that about 438 of those are truly from natural populations, and we're working to accession all of those into Geom. And so just to zoom out and sort of see where Geom fits, There's a great tradition. I think the genetic community was one of the first to have a tradition of publicly archiving genetic data. And so the United States, European, and Japanese genomic databases came together in this INSDC collaboration. And there's also been a fairly good tradition of archiving data, species occurrence and ecosystem level data in databases like GBIF and OBIS and Barcode of Life. But there's not been very good communication between databases that archive at this level of biodiversity and databases that archive at the genetic level of biodiversity. And such that only about 20% of potentially biodiversity relevant biosamples in the SRA have geospatial metadata attached to them. So Geome sort of provides a bridge between these different levels of biodiversity databases. And it's an agile sort of bridge. It's not run by any government body or anything like that. And allows us to sort of adapt to the changing data landscape. Okay, so I think we did actually very well with our open access population genetic database. We then also had these capacity building workshops in Indonesia, the Philippines, South Africa, Fiji, and then Libby Liggins took this and spun it off and actually created her own national network of marine biodiversity scientists in New Zealand called the Iramoana Network. And she had her own, she got that funded and had her own set of workshops around that. And then of course we had this distributed datathon. I'm not going to say much more about our workshops other than each one of them was tailored to the needs of our hosts. So for example, in Indonesia, they wanted to learn a lot about just how to use the command line and how to use all of these different analysis programs that were out there for their work on population genetics. South Africa wanted something a little bit more advanced, and we did a whole workshop around seascape genomics. Fiji, there's only one lab that does any sort of genetics out there. They wanted to work on species level stuff, and so we brought in some people to teach them about biodiversity informatics and got over 150,000 accessions added to GBIF. So we sort of met them where they were. And this is really what I sort of want DipNet to move forward into the future as being a capacity development organization for the Indo-Pacific. And then we have, we had a number of questions about the patterns of marine biodiversity at multiple levels of organization, looking at conservation units and looking across communities at their phylogeography. Okay, so I hope I've sort of given you a taste for how DipNet has addressed the size, the diversity, the political complexity, and the lack of cooperation, at least in its first 5 years of existence. And we're going to take a little break for just maybe 5 minutes. I'll look at the time, how we're doing. And then we can come back and talk about some science. Having been stuck at home myself, I know you probably have to go restart your kids' Zoom or get a snack or go to the restroom, but I'm also happy to take questions for about 5 minutes and then I'll get going again.
Anyone have questions?
I have a quick question.
Yeah.
I'm wondering if the database that you set up also has like a place for it to say if like the specimen itself was ever put into a voucher for museums.
Yes, we have we have basically, if you've ever looked at the Darwin Core site, we have all of those terms in there. So we can take a URI for a voucher, or if you have a museum accession number that you just want to put in there, we can take all of that.
Thank you.
I was just wondering to get I guess, a point of clarification. You mentioned a difference between two kind of sets of genetic databases. Do you mean like the very, like, I guess, open-ended databases that have species from all over the globe versus databases that are specific to Indo-Pacific region?
Yes, so basically, so Geom has become, through our collaboration with John Deck, Geom has become a global database. So where DipNet is just one team or project within Geom. So we're one group, we're the first group that we developed our own metadata standards, and we've stored our metadata in Geom. And in fact, you can store mitochondrial sequence data as text directly into Geom. Whereas, second and third generation sequencing data are stored on GenBank. So it's a global database now. I'm not sure if that answered your question or.
Yes, it did. Thank you.
I have a question. In the context of the political diversity that you mentioned in the Indo-Pacific, you also sort of seem to touch on your success in so streamlining perhaps permits and access and such things. I'm interested in that because, with the Nagoya Protocol allowing countries to be very, very restrictive about access and about taking specimens out of their countries and so on. I wonder whether that was the sort of context that you were thinking about when you tried to streamline things and
I might have missed the end of your question there, but yes, if you go to the Geom website and read through the data usage agreement that we created, it basically it says that in very nice lawyerly terms that we're just tracking the metadata for genomic data that are publicly available anyway, and every country retains their rights to I'd just invite you. It's so hard when you get into lawyerly language there, but it basically tries to protect the scientists while also protecting the rights of the country, and allowing them to still have the right to profit from the next cure for cancer or whatever that's created from that genomic data, while still but also facilitating open science and open data. Tries to walk that line. We haven't. That's a feature for DipNet is I imagine of creating a website where we really try to make clear the permitting requirements for each country. But that's a lot of work, and that's in the future. Okay, so there's a lot of science I want to talk to you about too, so let me move on. Okay, is that everybody can see that? Okay. All right, so now we're gonna talk some science, FST and other things. Just to start, very basic. Sewell writes FST is the standard population genetic way for defining population differentiation, genetic differentiation. So if you have two alleles in a two allele system, if they're equally distributed across two populations, you'll have FST of 0. And if they're completely segregated between populations, you'll have FST of 1. And we can get an analog of FST by looking at how heterozygosity in the total population is relates to the heterozygosity in the subpopulations. And in 1998, about the time I was in Moorea, Robin Waples wrote a very influential paper called Separating the Wheat from the Chaff, where he surveyed all of the genetic work that had been done in marine species and showed that in the vast majority of them, FST was well below 0.02, or the median FST was about 0.02. He looked at that and he said, well, the amount of sampling error that we get just from sampling 25 samples per population is about 0.02. So for marine species, you really can't differentiate the signal from the noise. And with that, whereas you can with anadromous or freshwater populations, and with that, he promptly went off and worked on salmon. That's kind of a joke. But those of us who are still working in the marine realm have named that area of parameter space after him, the Waples zone. So in regions, if we have effective population size across here on the x-axis, and proportion of migrants, that's the proportion of individuals migrating between populations on the y-axis, and FST is shown here in black. So most marine species inhabit this area of large effective population size. If you just picture the number of snails, intertidal snails that I studied for my dissertation across all intertidals across Hawaii and the Indo-Pacific, there's literally probably billions of them. And with effective sizes in the millions. And their larvae facilitate relatively high rates of migration, so that with FST very, very, very close to 0, it can be very difficult to differentiate between very well-connected small populations or large populations that are actually demographically independent of each other. So that's just to sort of outline the problem. We took this large database that we had put together for DipNet, and we went and we wanted to test some biogeographic hypotheses. We thought, okay, we've got this huge database of species. We can, and then we've got all of these hypotheses about how the Indo-Pacific is broken up biogeographically. And of course, we started with Brian Bowen's and Jack Briggs' 2012 Indo-Pacific provinces based on fish endemism. Then we looked at Keith et al.'s, they looked at range ends of coral species and came up with these provinces. Kulbicki et al. Did an area cladogram analysis on fish species and came up with a 2-level hierarchy of realms and provinces. Spalding did the marine ecoregions of the world, and they had a lot of different inputs, but it was basically sort of expert opinion about what made these different ecoregions distinct from each other. And they had a 3-level hierarchy of ecoregions, provinces, and realms. And then Charlie Varon had a sort of an area cladogram analysis on corals, and they came up with their own set of regions. And this is a lot of color to look at, but you can see that they clearly are not in complete agreement with each other. We just look at Brian's hypothesis. He has the whole central Indo-Pacific from the French Polynesia over to India as one single region, whereas For example, the Spalding realms have actually 3 fairly equally sized regions. And we thought, well, we can test these with genetic data by using analysis of variants or analysis of molecular variants called an AMOVA. And we worked with Peter Smouse and Chris Bird, and I developed this Bayesian information criterion where we just modified AMOVA a little bit and changed it so that we could test hypotheses that had different numbers of regions in them. So that you know, so we were penalizing by the number of regions, and so we can test across different hypotheses that have different numbers of regions. And you ready for the big reveal? Here it comes. We got a lot of noise. This is relative probability of each hypothesis. So the hypotheses across the bottom, and 56 species that passed our criteria for inclusion in the study across the y-axis here. And the heat is relative probability of each hypothesis. And you know, there are clear winners in a few cases, but it was fairly different across each species. So each species had its own hypothesis that it preferred based on the genetic data that we had. Now, a lot of this could have been done to the lack of co-sampling that I mentioned earlier. There's a lot of reasons why we might not have gotten this, but there is one little pattern that you can sort of see, which is that these hypotheses are arranged by number of regions. And I've already said that I think we mathematically corrected for the number of regions, so that shouldn't be an issue. But in general, the data seem to be favoring hypotheses that have a low number of regions. And if we just look at which hypothesis was favored for which species, and we look at it across phyla or across families within the vertebrates, it's just very noisy. There's 2 hypotheses that have the most support, Brian's hypothesis and the Spalding realms. There's no taxonomic pattern that I can see. So there's just a little bit of signal there that we can move on with. And that's, honestly, that's pretty much what we were expecting. That's par for the course in marine population genetics, is we just have all of this very low signal and a lot of noise. But we took these 2 hypotheses, which are somewhat different from each other, and we took each of the 5 barriers that they tended to support. So Spalding et al. Suggests that there might be a barrier right here between Indonesia and the Indian Ocean. And Brian's hypothesis says that there's a barrier between Hawaii and the rest of the Indo-Pacific. We looked at all those 5 barriers, and we broke it down into just those 5 barriers. And then we measured FST across each of those putative barriers. And let me walk you through this slide. Each of these dots here is the median FST that we've measured for this number of species across each one of these barriers. And you can see that we're clearly operating in the Whipple zone, right? The median phi-ST for each of these barriers is right around 0.025. So that's really, really tiny. We did a randomization, so we just randomized and bootstrapped the confidence intervals. And this barrier between the Indo-Pacific, between the Indian and Pacific, I should say, the Indo-Pacific barrier came out as significant. The barrier between Hawaii and the rest of the Pacific was significant, as was a barrier in the Central Pacific, between the Central Pacific and the Eastern Pacific. The other 2 barriers were not significantly greater than our random expectations. But the most interesting part for me was that I then did some really simple-minded coalescent simulations where I did these simulations in the Whipple zone. So I did really large effective population sizes of 500,000. So one population, a single Indo-Pacific population of 500,000 is allopatristically separated during the last glacial maximum about 20,000 generations ago. And that separation is continued. So there's no gene flow subsequent to that. When I did this really simple simulation with large effective population sizes, this is the simulated FST that I got back. And it's right in line with what we see across all of these other putative barriers. So FST is really low, but it's exactly what we would expect if there had been no gene flow across this barrier since the last glacial maximum. So what we could be seeing here are really, actually, possibly fairly robust barriers, for all we know, based on the FSTs that we're measuring. It's just that the populations are so large that FST is still very small. And so that was fairly eye-opening. So we can't distinguish between whether there is ongoing gene flow. There might be ongoing gene flow, but we simply can't distinguish that. But it could be that gene flow is low or negligible across these barriers. Okay, so we're clearly operating in the Whipple zone in the Indo-Pacific. Nothing has changed since 1998, and that's that's what that paper really identified very well. In 2006, ooh, I'm getting close. All right. In 2006, Robin Waples wrote another paper where he tried to identify what is a population. And he did a bunch of simulations where he looked at different methods and can they detect population structure and reject panmixia? And can they estimate the true number of populations? So just to remind you, FST, when it looks like this, when you're looking at allele frequencies, FST of 0 looks like equal distribution of alleles. FST of 1 is where alleles are completely segregated. And what Robin didn't do is he didn't simulate into the Whipple zone. What I've found, though, is that if we don't just consider the allelic distribution, if we also consider the coalescent history of these samples, that we can actually learn a lot more than just than we would just from FST. So if we reconstruct the allelic, the coalescent history, or if we integrate across all possible coalescent histories, what we actually find is that if this is the true history of these samples, then if we do a quick little parsimony calculation, and these are the locations where the samples were taken, then the ancestral sample came from population 2, and we can infer that there was a gene flow event from population 2 to population 1. And there was a second gene flow event from population 2 to population 1 in the white clay. So by looking at the relationships of the alleles, we've learned about the directionality of gene flow, as well as allowing us to create an actual metapopulation model of gene flow. So what I did was I simulated into the Whipple zone. I created a set of simulations of large effective population sizes of 10,000, 100,000, and 1 million, and fairly large levels of gene flow, leading to effective gene flows of 10, 100, and 1,000 migrants per generation. So simulating into the marine area, into the Whipple zone. And then I took a coalescent sampler that uses the method that I just discussed called migrate. And migrate also allows us to test hypotheses in a likelihood framework. So I took these simulated data and I tested them under a hypothesis of a stepping stone model, where every population has a different size and migration parameters are all different from each other. Tested another population model where there is regional structure. So each of these populations is actually a single population, the ones in boxes here. So we're not able to detect every species, basically, or sorry, every population. I tested the hypothesis, which was the true model that the data were simulated, which is that all populations have the same size and are exchanging migrants at the same rate. Tested a variant of that, again, with regions, as well as an island model where all populations are exchanging migrants at the same rate with different numbers of regions, as well as tested the hypothesis of panmixia. And these are the results I got. The probability of each model is shown in the stacked bar. And you can see that for effective migrants of 10 effective migrants per generation, Migrate gets the correct model almost 100% of the time. For 100 migrants per generation, and this is what blew me away, Migrate still gets the correct model almost 100% of the time, except in this model with effective size of 10,000, in which case it still gets almost the correct model. It just tends to see fewer numbers of regions, or sorry, fewer numbers of populations. For effective migrants of 1,000, okay, nothing. It's not able to reconstruct that. But this right here is way, way better than FST can do. So Rob was nice enough to let me have access to all of the hard-won data of the Tobo lab, 41 species across the Hawaiian archipelago. And just really quickly, across that archipelago, just as a first order sort of estimation, we can expect somewhat stepwise population. You know, most larvae are being exchanged if they're being exchanged at all, they're being exchanged between neighboring islands. Of course, it's not perfect. There are longer distance migration events, but a stepping stone model is a good first approximation. Kim Selko did an analysis of these data in which she found that 20 of the species, these numbers don't sum to 41. She had slightly fewer species to work with, but 20 of them had regional structure. So there was two or three panmictic regions within Hawaii. 6 of them were completely panmictic, and 4 of them showed a pattern that was consistent with a stepping stone model of dispersal. 7 of them were chaos, sort of the term that marine population geneticists use when our genetic data make no sense whatsoever. I went and put these data into Migrate and tested the same sort of set of models that I did with the simulation. So I tested a hypothesis of panmixia, a hypothesis of high islands versus low islands, another hypothesis with a break at French frigate shoals, another regional hypothesis with 3 panmictic regions, with another break at Lisiansky Atoll, an island model where all islands are exchanging migrants with each other, and then the stepping stone 2-parameter model where you have equal migration between all islands and equal population sizes. And then the full model where all islands can have all populations can have different sizes and different rates of gene flow. And these are my results. This is a very noisy graph, but it's the same graph that I showed you with the simulation. So this is the relative probability of each model. And what you can basically see here is that 18 of the 26 species came back with some clear model that it chose, whether that was panmixia or the full stepping stone model or the 2-parameter stepping stone model. And most interestingly, notice that it picked the full model. So it picked what our understanding of nature is, which is that every population is of a different size, and migrations are migration rates are not all different, are all different. So it's not and I already know that it's not overfitting, because when the true model is each population is exchanging different sizes, each population is exchanging the same rate, that's what migrate chooses. So migrate is not overfitting, it's choosing the most complex model because that reflects reality. And if we contrast my results to those of Kim, I actually found the model that meets sort of our a priori expectations about these species much better than using a traditional FST inference does. So 18 of them were stepping stone, 4 were still, the hypothesis of panmixia was best. However, 15 of them gave us an ambiguous result. I would argue, though, that it's better to have sort of an honest, ambiguous result than to try to shoehorn it into one of several models. That at least the method is saying, well, I don't know. I'm not gonna try to pick any particular model. I'm just going to say, I don't know, and we need more data. Okay, so I kind of rushed through the end of that because I know I'm coming up on 6:30 or 12:30 your time. But I hope I've sort of given you a taste now for how we've addressed these low expected values for FST estimators using coalescent methods, how we've addressed the problem of large effective population sizes and high migration rates. In conclusion, I think that science in the Indo-Pacific is only going to move forward when we can build capacity in Indo-Pacific countries, developing Indo-Pacific countries, to do the science themselves and to study their own biodiversity. And we need to do that through collaboration and the open data environment that's created by DIPNet. Our reanalysis with 56 Indo-Pacific species supports bioregionalizations with few regions, and we have more analysis about genetic diversity to come. Our coalescent models do have very high power to detect a stepping stone model with effective migration up to 100 individuals per generation. And in a model selection framework, these coalescent methods have really high power to tell us about the number of demographically independent populations. So I would say that this work was possibly one of the first to suggest that each Hawaiian island could be managed separately. It's possibly demographically, the populations on each island are demographically independent from the other islands. And with that, I just want to thank all the people that were involved in this research. And I know I'm right up against the end here. So any questions are certainly welcome.
Great, thanks very much. So I think some people need to jump off the call to get to another meeting, including me, so I will leave. But if there are questions, hopefully y'all can have a chat about that. So thanks very much.
Thank you.
Any questions?
I've got one question. How much computation does it take to do those coalescent, all the coalescent simulations?
A lot. Yeah, that took me. I had a few false starts with it. I think it took me at least in the final run with all of those species, took me at least 6 months, I want to say.
Well, 6 months.
That was across. That was using the Hawaii cluster.
So 6 months of the cluster crunching away.
Yep. Yeah, and we had to work with. we had to work with the HPC on campus because they don't allow processes to run that long. So we actually had to buy our own condo and take off the time limits because otherwise it couldn't complete within the time limits that they were willing to do.